If you are ambitious and you have a vision and you are determined to execute, believe me, you will be successful in all your goals. Today I am going to move on to the subcutaneous nematode, which is Dracunculus medinesis. Now there are several names given to this parasite. It is called guinea worm because this was first seen in Guinea coast of West Africa. It is also known as serpent worm, and it is known as the fiery serpent, which is mentioned in the Bible. And then we call it Medina worm because it was detected in Medina as well. So these three names you are asked about in your exams, and you should be familiar with all the names which are given to Dracunculus medinesis. Now, the learning objectives are that at the end of the session, all of you should be familiar with the different names which are given to this parasite. Then you should be able to give the lab diagnosis, how you're going to diagnose this organism in the laboratory, what are its clinical manifestations, and the pathogenesis. You must be familiar with the two types of host involved in this parasite. There's an intermediate host and a definitive host. Man is the definitive host and the intermediate host is a Cyclops. Then you should be able to draw and label the life cycle in detail. And there are other terms which are being used with this parasite, and this is viviparous. Viviparous are the parasites which are just going to produce the embryos or the larval form of the parasite, and they are not going to produce eggs. Oviparous are the ones which are going to, these are parasites which are going to produce eggs and not the larval forms. And we have already done the oviparous like Ascaris lumbricoides or like Ankylostoma duodenale. And the viviparous is Dracunculus medinesis, which is not going to lay any eggs, it is directly going to produce the embryos or the larval forms. Coming to your examination question, generally this is the type of case history which you will see in your exam. A 52 years old washerman presenting with history of urticarial rash, and you can see this blister on the foot which ruptured, resulting in formation of this ulcer. And from the center of the ulcer you can visualize this white parasite which is coming out. This white parasite which is coming out is the worm or Dracunculus medinesis. If you take the fluid from this blister and examine it under the microscope, you're going to see these coiled embryos, and besides the coiled embryos you will also see a large number, with the coiled embryos you are also going to see these eosinophils and polymorphs. If you culture the fluid, you will not be able to isolate any bacteria from it, so it is sterile fluid. Coming to the questions associated with this part, now which nematode is responsible for this condition? What is the pathogenesis of blister formation in this case? How is the parasite diagnosed? Or what is its life cycle? Or draw and label its life cycle. Coming to its habitat, I have already told you that this parasite is viviparous, which means that it is just going to produce the larval forms. And the adult female is present in the subcutaneous tissues, especially the legs, the feet, the arms, or the back. Man is the definitive host, and the intermediate hosts are the cyclops or the copods. Now, definitive host is the one which is going to harbor the sexual or the adult form of this parasite. Intermediate hosts are the ones which will have the asexual form of the parasites like the larval forms. The morphology, this parasite is the largest nematode. Now we have, while we were doing Ascaris, we said Ascaris is the largest intestinal nematode. However, when you get an MCQ on the largest nematode as such, then the largest nematode is Dracunculus medinensis. This is a subcutaneous nematode and it is the largest nematode. It is as long as a meter in length, and if you look at this slide, it is milky white in color, and this is the anterior end, which is rounded, and the posterior end is tapering. And I've been repeating this again and again. This is one of your viva question as well, and you get this question as part of your OSPE. It is a viviparous parasite, which means that it is producing larval forms and no eggs are produced. No eggs are produced by this parasite. The embryos are coil structures. Here you can see the anterior end, it is rounded, and the posterior end or the tail is tapering, and they are discharged when the affected part or the ulcer is submerged in water, and the gravid female is going to just push them out in batches for 3 weeks till it empties its uterine cavity completely. So they are coiled structures. The anterior end is rounded, the posterior end is tapered, and they are only discharged when the ulcer is submerged in water. When it comes in contact with water, then these embryos are released in the water. The body fluid which it has is very toxic, and when it comes to the surface of the skin, it is going to produce blisters, and these blisters are responsible for the ulcer and the pathogenesis associated with this pathogen. Coming to its life cycle, as I've been telling you, life cycles are very important. In this case, man is the definitive host, and the intermediate hosts are the cyclops or the copods. The cycle starts with development in man. Man gets infected, as you can see in this picture. This female is collecting water from the pond, which is infected with the cyclops containing the larval forms. And here you can see this man is directly drinking water from the ponds. Now, the water which is collected has cyclops, and these cyclops have larval forms in them. When this water goes into the stomach, the digestive juices are going to break down or digest the cyclops, and this results in liberation of the larva. The larva is going to pass through the stomach and the intestinal mucosa, and it goes and lodges itself in the retroperitoneal connective tissue. Here they are going to develop into mature forms or adult forms, and after 6 months the gravid female is going to move to the subcutaneous tissues, which will eventually come in contact with water, like the back of water carriers. Now, in the ancient times, men used to carry water on their back from one place to the other. So, this parasite would be seen here at the back of these water carriers, or they are seen coming out of the feet or legs of washerman, as you can see over here. Now, initially, when it comes to the surface of the skin, under the surface of the skin, it is going to release a toxic fluid, and this toxic fluid is going to provoke a blister formation. So these blisters are formed when this parasite comes under the skin and start producing the toxic substance. The blister eventually breaks down, as you can see over here. The blister is inflamed and it breaks down, results in formation of this ulcer, and from the center of this ulcer you can easily visualize the worm coming out. And this worm, when it comes out and when this ulcer comes in contact with water, it is going to discharge this milky white fluid. You can easily make out this milky white fluid which is being discharged by the female. The head of the female is coming out of the center of the ulcer, and it is going to release thousands of these coiled motile embryos. So this is how these embryos come out. When this ulcer comes in contact with water, the female is going to discharge this milky white fluid, and this milky white fluid is full of coiled embryos. Second stage of this life cycle is development in the cyclops. or the copepods. Now, when the lava are discharged in water, they are ingested by the cyclops, which are there in the ponds, and they are going to break through the intestines of these cyclops and comes to lie in the body cavity, where these larval forms are going to mold twice and they become the infected form. And this infected form larvae is known as L3, or the third larval form. And now these cyclops containing the infected larvae are present in the water. So humans become infected when they drink the water from these contaminated pods having cyclops with the larval forms. So this is the life cycle of Dracunculus medinesis. When in the exams you're asked to draw and label the life cycle, then this is what you're going to show us. Humans are going to, sorry, humans are infected, as I said, with when they drink water which is contaminated with copepods containing the larval forms, the L3 forms, which are the infected forms. These larval forms are released from the copepods in the stomach and intestines when they are acted upon by the digestive juices, and they are going to penetrate through the intestinal wall or the stomach wall into the retroperitoneal connective tissue. There they are going to mature into adult form, and these are the adult form, and they are going to the male fertilizes the female, and the fertilized female is then going to migrate to the surface of the under the surface of the skin, to the part which is going to finally come in contact with water. Now when it comes in contact with water, the blister which is formed here is going to break down, and this the head of the female comes out, and it's going to discharge the larval forms in the water. And these larval forms are the L1 forms, which are the immature form of the larva. The copepods or the cyclops are going to digest or consume this L1 larvae, and within this the cyclops, the larva undergoes two molds, and they are developed into the infected form of the larvae, which is L3 larvae. Now these copepods or cyclops in the water are then again taken up or consumed by males, or by humans, and the cycle is repeated again. As the cyclops goes into the stomach, they are again acted upon by the digestive juices, and the larva comes out, and it matures into adult form, moves down, forms a blister, and the life cycle is repeated again. Coming to the clinical features, the clinical manifestation is the most important, is the formation of this blister. Now, as I told you, this blister is formed just beneath the skin when this parasite releases the toxic substance, and this results, and you can you can observe the inflammation around this the blister. And when the fluid is sterile, and when this blister breaks down, it results in formation of this painful ulcer. This is a very painful ulcer, and from the center of the ulcer you can see the head of the female coming out. And here you can again see that the parasite is being pulled out from the center of the ulcer. Here this picture is showing you inflamed ulcer, and again you can see the white parasite coming out. So there could be systemic symptoms. The systemic symptoms are the generalized symptoms which are associated with this parasite. There could be erythema, there could be urticarial rash, the patient may present with nausea or giddiness. The local lesions are very important, which is in form of blisters, and these blisters have indurated inflamed margins, and they are very painful. These ulcers can be a single ulcer on the ankle or hands or the arms or the trunk, or there may be multiple ulcers. If you remove the fluid from this blister, it is sterile, which means that it is not going to it does not contain any bacteria and will not produce any will not be isolated in the lab. Bacteria will not be isolated in the lab. It is yellow in color and it contains monocytes, eosinophils, and most important, numerous embryos. Coming to its lab diagnosis, it is again like with other nematodes, there could be direct evidence or indirect evidence. Direct evidence is detection of the adult worm, which can easily be done, and because the gravid female is coming out of the center of the ulcer, so you can just twine it around a stick and you can pull it out gently. As you can see over here, a large portion has been pulled out, and if this sperm dies in the subcutaneous tissue, then X-ray would show calcified worms. This is again a picture showing you the worm being pulled out, and you can see the length of the worm which has been pulled out here and here. Second is detection of the embryo. So if you put a drop of cold water on the ulcer, milky white fluid will be released. As you can see over here, this fluid is red, and if you do an microscopic examination of this milky white fluid, you're going to see a large number of these larval forms and eosinophils as well as polymorphs. Then we have indirect evidence. Indirect evidence would be like indirect evidence is immunological test, because this parasite is moving in the body, so it is activating your immune system to produce antibodies. So antibodies to Dracunculus medinesis can be detected by using various serological or immunological tests like ELISA or fluorescent antibody techniques, etc. Blood examination is going to show you marked eosinophilia, and if you perform a skin test which is known as intradermal test, you inject the antigen intradermally, and if antibodies are present, they are going to come and get attached to the antigen, so resulting in antigen antibody formation, and there is going to be wheal formation within 24 hours. So with this we are done with parasitology. We have done the whole of parasitology, the important parasites which all of you should know. So the assignment for you today is an SCQ, which you're going to give the answer within 24 hours, and it should be handwritten. A washerman presents with a blister on his foot, which is highly inflamed, very painful ulcer, and a whitish worm is coming out. Which nematode is responsible for this condition? Give the pathogenesis of blister formation. How is this blister formed? How is this parasite diagnosed in the laboratory? Thank you very much.